Okay, good morning. All right, I think we finished this slide last class, right? And we talked about glycolytic pathways and gluconeogenic pathway. And these two pathways is kind of, an opposite in terms of its function. One, like glycolysis, basically is active during the fat condition, and the purpose of glycolysis is to oxidize glucose and produce energy. And whether the glycolysis glucose will be completely oxidized to CO2 water or just partially oxidized to lactate, pyruvate and lactate, depend on the cell condition, right? Whether cell is under the aerobic condition or anaerobic condition, and the gluconeogenesis. The main function of this pathways is to produce glucose during the fasting when we don't have when we are not able to get glucose from the diet. So our body synthesizes glucose from non-sugar precursors, right, to glucose, and the main tissue that does gluconeogenesis is the liver and kidney. When the gluconeogenesis, we're going to talk about later a little bit more about gluconeogenesis in the kidney. And the key pathway, the key enzyme that I ask you to remember, right? The three key reactions in the glycolysis, and four actually key reaction or really limit reaction in the gluconeogenic pathways, and the key enzyme, right? You want to remember. And also I mentioned that in the gluconeogenesis, whether the four key reaction involved or three key reaction or two key reaction involved in the gluconeogenesis depend on which type of precursor, right, the cell used for gluconeogenesis, if you still remember. And two key reactions occur in the mitochondria, and other two key reactions in the cytosol. And we also mentioned the two important, the gluconeogenic cycles. You still remember which two cycles that we learned for gluconeogenesis? Can you tell us? Cori cycles, right? And the alanine cycles. So Cori cycles basically use what is the precursors for Cori cycle for gluconeogenesis? Remember Cori cycles? Lactate, right? Lactate is substrate or precursors for gluconeogenesis. And where does lactate come from? Which tissues contribute? Muscle and red blood cells, right? Okay, and alanine, of course, cycles. Alanine is the precursors, and where does alanine comes from? Muscle, right? Muscle is builds alanine for gluconeogenesis in the liver. Okay, any question? Now let's do some exercise and something related to the clinic. PEP-CK is a key enzyme, right, in the gluconeogenesis. Pyruvate of phosphoenolpyruvate carboxykinase, right? Carboxykinase. PEP-CK is one of the key enzyme in the gluconeogenesis in the cytosol, right? Remember the reaction? Okay. So if someone has the defects in the PEP-CK enzyme, again, it's a key enzyme, the gluconeogenesis. And what happens to blood glucose control, particularly during the fasting condition? the fat condition, well, okay, right? We can get a glucose from the diet, so we'll be able to maintain blood glucose level. But during the fasting, gluconeogenesis is very important, right? To make our body, have a, to prevent low blood glucose or hypoglycemia happen. So what happens to the blood glucose control if the person doesn't have enough active PEP-CK, the key enzyme? Yeah, hypoglycemia, right? We're not able to maintain the blood glucose level. So blood glucose level will dropped during the fasting. So it's not good idea, for this kind of patient, if to experience the fasting for a long time, right? And the second question, what happens to blood lactate levels? Sorry, hypoglycemia, right? And what happens to blood lactate levels? Increase? Yes, because lactate cannot be used for gluconeogenesis, right? Because of the lacking of this enzyme, so lots of lactate will be accumulated in the circulation, so causing the lactate acidosis, right? Again, this will usually happen during the fasting condition, right? And last question, what happens to glycogen store? Remember during the fasting, which two glucose metabolic pathways are active and to contribute to circulating glucose levels? Gluconeogenesis is one, right? And what are the other pathways that also contribute glucose to the glucose in the circulation? Glycogen breakdown, right, in the liver. So, so what happens if gluconeogenic pathway cannot be active during the fasting? What happened to the glycogen breakdown pathway?
It
Decreases? Are you sure? I mean,
the glycogen?
right. I mean the pathway. Sorry, you're talking about the glycogen store.
Yeah.
Decrease. Yes. I mean, you're right. I was asking, whether the glycogen breakdown pathway activity will be increased. Yes, right. And you're right. And glycogen, and this particular question talk about glycogen store, right? So glycogen store will be decreased, right, during the fasting in the patient, again, with defect in the PEP-CK, the key enzyme in the gluconeogenesis. Right. So glycogen depletion, right? They use a lot of glycogen to compensate, the defects in the gluconeogenic activity, right? That makes sense? All right. Now let's talk about the other key enzyme, glucose 6-phosphatase. It is also one of the key enzymes in gluconeogenesis, right? The catalyze the last steps of a reaction in the gluconeogenesis, right? Converts glucose 6-phosphate to glucose, right? And release glucose. So what happens to the blood glucose control in a patient with defect in the glucose 6-phosphatase? What happened to the blood glucose level during the fasting? decrease similarly, right? So if the patient with defect in the glucose 6-phosphatase will also experience a hypoglycemia during the fasting, right? That makes sense. And what happens to blood lactate levels? Yeah, similarly, right? Similarly increase the lactate level in the circulation. And interesting, what happens to glycogen store in this patient? The patient had a deficient, glucose 6-phosphatase deficient. And this is a key enzyme, right? That catalyzes last reaction and convert glucose 6-phosphate to glucose. You don't know? Glycogen should be accumulated, and you think accumulate, right? Yes. So can you tell us what the reason why the glycogen will be accumulated in this particular patient?
So that's the enzyme that's responsible for glycogen breakdown?
Yes, great. So glycogen breakdown, the glucose 6-phosphatase not only is the enzyme that involves the last reaction, right, in the gluconeogenesis, but also as in the glycogen breakdown. Glycogen breakdown, it's also involved the steps that glucose 6-phosphate converts to glucose. It's also the last step reaction in the glycogen breakdown pathway, right? So glucose 6-phosphate will accumulate if we don't have a glucose 6-phosphatase. So glucose 6-phosphate is a substrate that can inhibit glycogen breakdown enzyme, which we're going to talk about later, right? But just let you know, this is the very strong inhibitors of a glycogen breakdown enzyme. So when glucose 6-phosphatase is lacking, then glucose 6-phosphate cannot be converted to glucose and accumulate, right? And then inhibit glycogen breakdown. So that leads to, increase the glycogen accumulation in the liver. All right, so let's just give you some idea, the pathway that involve not only okay. This is the this gluconeogenic pathway, right? So glucose 6-phosphate phosphate converts glucose. This is the last reaction in the gluconeogenic pathway, but it's also the last reaction, the glycogen breakdown pathway, right? So this is a common pathway for both gluconeogenesis and glycogen breakdown. Okay, now let's talk about the gluconeogenesis in the kidney. So kidney can also make glucose and contribute the 10% of gluconeogenic derived glucose. And the activity of gluconeogenesis in the kidney depend on the duration or time of the fasting. So if only fasted for a couple hours, then liver will be the most active tissue that does the gluconeogenesis, right? But when the fasting is prolonged or continuous, then kidney also start to make glucose. So for example, after 14 to 16 hours fasting, and 20 to 25% of glucose neogenically produced glucose, right, from actually kidney. So kidney contributed 20 to 25 glucose during the prolonged fasting. And if fasting goes longer, usually like 24 hours later, right, and 50% of gluconeogenic, glucose from gluconeogenesis actually coming from the kidney. So, and the other important thing is that most the precursors for gluconeogenesis in the kidney mostly from the amino acids, right? So amino acids are the main precursors for gluconeogenesis in the kidney. And last important point in the gluconeogenic kidney is that the TCA cycles is actually involved in the gluconeogenesis. We're also going to talk about next slide. It's called anaeroprosis and a process reaction. That means, the reaction that involved in the TCA cycles. So 3 points, right, that important that you need to remember related to gluconeogenesis in the kidney. First is the timing, right, when only prolonged fasting after longer than 14, 16, or 24 hours, and kidney become important gluconeogenic organs. And secondly, the precursors mostly are the amino acids, right, for kidney gluconeogenesis. And lastly, the TCA cycles, the anaeroprosis and keroprosis reactions that are also involved in the kidney, in the gluconeogenesis in the kidney. Okay, now let's talk about anaeroprosis and the keroprosis. So this is actually a number of the enzymatic reaction or pathways that can repress the pool of so-called intermediates in the TCA cycles, right? TCA cycle we know had lots of many reactions and produces lots of intermediates, right? And this intermediate concentration has to be maintained in the normal range, otherwise the TCA cycle cannot function. So how we can maintain the concentration of intermediate in TCA cycles, and that is a function of the anaeroprosis and keroprosis. That means that the other reactions that connect to the TCA cycle and can provide the intermediates, right, to refresh the intermediates, or if we also need to prevent the accumulation of intermediates, so some reaction can remove the intermediate from the TCA cycle. So adding intermediate to TCA cycles or remove the intermediate from TCA cycles. So then can cell be able to maintain the right concentration of intermediate to make TCA cycle functioning, right? So this is just in general ideas, and specifically the anaeroprosis reactions means that the reaction that can add the intermediates into the TCA cycles, right? And keroprosis reactions, that reaction can somehow take the intermediate away from the TCA cycles, right? And the purpose of this anaeroprosis and keroprosis is to maintain the normal concentration of the TCA cycle intermediates and to maintain the normal function of TCA cycles, right? If you look at this slide, I just get all the, oops. So for example, the citrate, citrate is one of the TCA cycle intermediates, right? And citrate usually can be taken away from the TCA cycles and be converted to acetyl-CoA, and the acetyl-CoA eventually can enter the fatty acid synthesis pathway for synthesis of fatty acids. So this pathway basically actually take the citrate away from the TCA cycles, right? So can cause the reduced TCA cycle concentration in this pool of TCA cycle. So and similarly for the oxaloacetate, so this is also the intermediate of TCA cycle, but this substrate can be used for gluconeogenesis, right? And so these two pathways or reactions is called, can you tell me, based on the definition I just described, it should be catalysis or anaeroprosis reaction? Catalysis reaction, yes. And the rest of that are highlighted in blue. Basically, you're adding, intermediate to the TCA cycles, right? For example, let me just take the glumine and glumate, right? And usually contribute alpha-keto group, right, to the TCA cycles, adding alpha-keto group into the TCA cycles, and similar for the other blue highlighted reactions, right? You're adding the intermediate to the TCA cycles to prevent the reduce of the concentration of this intermediate. So this reactions called anaeroprosis, right? Reactions, right? So seems kind of simple to the concepts, and then let's talk about the role of TCA cycle in gluconeogenesis, as I just mentioned, right? The kidney gluconeogenesis in the kidney that usually involved in the TCA cycles because the kidney uses amino acids as the precursors to gluconeogenesis, right? So it's just here. There's one example, if glumate as the precursor gluconeogenesis, right? So can convert it to alpha-ketoglutarate and go through the many steps of a TCA cycle and reaction in the TCA cycles and convert it eventually to oxaloacetate. So oxaloacetate then leave the TCA cycle and convert it PEP and enter the gluconeogenic pathway, right, for synthesis of glucose. So this is the the new gluconeogenic pathway that involve the amino acids precursors and mostly occur in the kidney, right? And of course, this anaeroprosis, right, and catalysis reaction also involved in the gluconeogenesis. Right? Does that make sense? Right? And let's see. And anaeroprosis and catalysis is also important. That involved in the glumine metabolism in the small intestine. If you still remember, I mentioned earlier, small intestine usually use some amino acids as the energy, right, and also glucose. So which amino acids that enterocytes of small intestine can use for energy? Still remember? No? Glumine, aspartate, right? The 3 amino acids. And so here just sort of example how small intestine utilize glutamate as energy. This is a pathway, for energy production. So glutamine is first converted glutamate and alpha ketoglutarate, and then goes through several reactions. The TC cycle converts to malate, and malate then leave the TC cycle and also enter the cytosol, and to convert to the oxaloacetate and PEP, and eventually to pyruvate. And pyruvate then re-enter the mitochondria, converted to, acetyl-CoA, and then enter TC cycle for complete oxidation. So kind of, a long distance, right, or something like this, to be eventually being oxidized and for energy. Okay, so, and this slide also talk about the role of anaerobic and ketosis in the glyconeogenesis in adipose tissue. So remember, the adipose tissue is the main tissue for the synthesis of triglyceride, right? And triglyceride synthesis requires two parts. One is glycerol backbone, and the other part is the fatty acids, right? And glycerol backbone, I think we talked about before that mainly synthesized from glycolysis, right, from glucose in the fat condition. And in addition to this pathway, and the amino acids metabolism can also contribute some, to the synthesis of glycerol backbone. So this is the pathways how amino acids can be metabolized and used for synthesis of glycerol 3-phosphate, which is the backbone, right, for triglyceride synthesis, adding 3 fatty acids, right, to the glycerol backbone. So amino acids can be metabolized to pyruvate. Just show you last slide, right? And the pyruvate enter the TC mitochondria and TC cycle, and can be complete oxidized, and also some of the pyruvate can be directly converted to oxaloacetate and to malate, and malate, enters cytosol to the similar pathway that I described before, right? And PEP can not only be used for glucose synthesis, but also glycerol phosphate synthesis, right? And you don't have to remember the all detailed reaction, but just know the general concept. The glycerol 3-phosphate or backbone can be synthesized from glycolysis, from glucose, right? And also can be synthesized from amino acids metabolism. Okay, so the third, I think the third, right? Yes. The third pathways of glucose metabolism we're going to talk about is pentose phosphate pathway. So pentose phosphate pathway is relatively simple. What you need to know is the substrate, starting molecules, right, which is glucose 6-phosphate. So glucose 6-phosphate can be metabolized through the pentose phosphate pathway, the substrate of a pentose phosphate pathway, and several reactions. And the purpose of a pentose phosphate pathway is to synthesize the 5 carbon sugar phosphate, right? So this is sort of a product of this reaction. And also during the several this pathway reactions, the NADPH is generated. So NADPH is required for the synthesis of fatty acids, ketone bodies, and cholesterol, right? And so this pathway is relatively simple and short, but it's also important, right? And the purpose, again, for the synthesis of 5 carbon sugars, and which is important substrate for synthesis of the nucleotide, like DNA, RNA, right? So it's important, and NADH is also important for the synthesis of fatty acids, the lipid synthesis. So this pathway is sometimes, it's considered a secondary pathway of glucose oxidation, and it provides the sources of a reducing equivalent, NADH, right, for fatty acid synthesis, and glucose, the ketone body and the cholesterol, and most importantly is the production of a 5 carbon sugar phosphate, and this reaction usually takes place in the cytosol. So as I mentioned, NADPH produced from this pathways is required for synthesis of fatty acids, cholesterol as sterols, right? And also NADPH is important for maintenance of the integrity of the red blood cell membrane. And ribose 5-phosphate is essential for biosynthesis of a nucleotide and nucleic acid like ATP, coenzyme, NAD, NADP, and FAD, and nucleotides, RNA and DNA, right? All right. Now let's talk about the regulation of glycolysis and gluconeogenesis. As I mentioned, that these two pathways is at the opposite function, right? And depend on whether which pathway is activated or deactivated, depend on the physiologic condition, whether under the fat condition or fasting. And we only wanted one of this pathway, being active, right? And we don't want the other active because they have opposite function. Does that make sense, right? So our body need to regulate under the different physiological condition to allow the one pathway active and the other pathways inactive, that you can efficiently metabolize glucose or synthesize glucose. Does that make sense, right? So, and how our body does this kind of regulation, there are actually several levels. You regulate this activity at several levels. First is glucose transport, and secondly, the so-called substrate cycles. So the intermediates of these two different pathways can regulate enzyme activities. And lastly, the hormonal regulation, like insulin, glucagon, right? Usually the levels are changed during fat fasting condition. So insulin and glucagon actually, I think is like the top levels of a regulation, right? And then the glucose transporter and the substrate levels of regulation. So the final targets of this regulation should be the key enzymes in each pathway. That makes sense, right? So when only you regulate enzyme activity, then you can control the whole pathway, activity, right? So just give you kind of general idea. And I just want to mention again, the hormonal regulation usually regulates enzyme activities and also regulates enzyme concentrations or gene expression, right? So glucose transporters that we already know, and I ask you to concentrate on this 5 transporters, and we know that GLU2 basically is more abundant in liver, pancreas, and GLU4 is more active in the muscle, right, including skeletal muscle, cardiac muscle, and adipose tissue. And the 2 new concept that I wanted to introduce here is called affinity for glucose and Km. Affinity glucose for glucose, that means the transport, how easily glucose can bind to the transporters, right? And can transport glucose. And and if you realize that the affinity and Km, they actually come opposite. For example, GLU1, they have a high affinity for glucose but lower Km. So I'm going to explain on the next slide why affinity and Km is kind of opposite, right? So the McKay's constant, Km, have you guys learned about Km before? No? Okay. So Km is usually defined as the substrate concentration at half the maximum velocity. So, for example, enzymatic reaction usually involve the 3 components, right? The substrate, enzymes, and the product. So whether this reaction can take place, how would I try to say? The speed of a reaction, how quickly this reaction take place, depend on the 3 things actually. 1 is the substrate concentration, and 2 is the enzyme concentration, right? And also the product concentration also control the speed, the reaction of the speed, right? So have you guys learned about this, right? So, for example, if we wanted to reaction, happen faster, what do we usually do? To increase the substrate concentration, right? And to add more substrate, and then we can increase the speed of reaction. And the other important thing is that the enzyme activity means that how easily that enzyme will work for the substrate, right? So so this is the diagram showing the substrate concentration. When we add the more substrate, and you see the speed of the reaction actually increases, right? But it has a kind of plateau. When reach some kind of point, then even you add more substrate, then the reaction wouldn't increase anymore, right? So so you have a sort of a maximum the speed of reaction, and this depends on how much substrate you add to the reaction. So the Km actually defined as the the half of maximum, how much the substrate is needed for the half of for the reaction to reach the maximum of speed. Does that make sense? Right. So so again, it depends, how easily the enzyme bind to the substrate. So this something is sort of affinity. If enzyme is more easy to bind the substrate, and the affinity of this enzyme for substrate is high, right? So if the enzyme, so there's two concepts, okay? The affinity is that how easy the enzyme, bind to the substrate. So it's the affinity for the substrate. And the Km is just when you increase the substrate concentration, you raise the reaction speed. So the define was, reached as the half or maximum reaction. So if the enzyme affinity for the substrate is high, then you will require less substrate for reach the half maximum speed. Does that make sense, right? So if the affinity of the enzyme for the substrate is low, that means you need more substrate in order to reach the half of the maximum speed. Does that make sense, right? So that's why it kind of been opposite, the Km and affinity. Shall we take a break? Okay, 10 minutes break. All right. Okay, let's move on. So now let's talk about the specific glucose transporters, about their Km affinity, so then can help us to understand better, how a glucose transporter regulates glucose uptake, right, during fasting. For example, GLU1 and GLU3 basically have lower Km or high affinity for glucose. So that means the activity of GLU1 and GLU3, activation of GLU1 and GLU3 does not require a very high levels of glucose, blood glucose, right? So that these two transporters are active when blood glucose levels are low because they have a high affinity for glucose, right? So they even have a lower glucose and they can be active and transport the glucose into the cell. So these two transporters basically is responsible for basal glucose uptake in most of tissues, right? And GLU2, we know basically it's best high in liver and pancreas, beta cell, and this type of glucose transporters basically had high Km and low affinity for glucose. So it means that you need to have a very high levels of blood glucose level, then GLU2 can be activated, right? So that makes sense, right? The GLU2 basically, particularly in the beta cell, only respond to the high blood glucose level after meal, then activate and transport the glucose. And GLU4 is a little bit different. They also have a high affinity for glucose. That means you just need a low glucose level. The GLU4 can be activated, right? But we know that GLU4 is insulin dependent, right? Only being active when insulin level is high, right? So the question here, why the basal levels, the GLU4 is not the transporter that responds for that's transported glucose under the basal condition. That means when insulin level is not high, although they have a higher affinity for glucose. Why GLU4 is not the glucose transporter that transport basal level glucose? Yes.
Because it's dependent on insulin, which is made by GLU2, which has a low affinity. So it kind of depends on that.
Okay, so I think the first part of your answer is correct. Depend on insulin, right? So when we don't have a high level of insulin, then GLU4 level on the cell surface is very low, right? Insulin stimulated translocation, if you still remember, GLU4 from inside of cell to the cell surface, right? So under the basal condition, that means when we have a lower insulin level and we don't have much GLU4 on the cell surface available for glucose transport uptake, right? So even you have a high affinity for glucose, but we don't have much GLU4 available on the cell surface without high level of insulin, right? So again, GLU4 is, it's the uptake of glucose depend on insulin. That makes sense? All right. So and the last point, just kind of review, I guess. You guys all know, right? GLU2 is regulated by glucose level, and GLUT4 is regulated by insulin level, right? Okay, now let's talk about the substrate cycle. How a substrate that in each metabolic pathways regulates the activity of each pathway. Glycolysis, if you still remember, we talk about the 3 key reactions, right? Rate-limiting reactions in the glycolysis pathways catalyzed by the 3 enzymes like glucokinase or hexokinase, and phosphofructose kinase and pyruvate kinase. And this 3 key reaction in glycolysis again is irreversible, right? Cannot go the opposite direction, catalyze, the opposite direction of reaction. So in the gluconeogenesis, you need to another enzyme to catalyze irreversible reaction. So in the gluconeogenic pathway, we have 4 key enzymes that catalyze the 3 irreversible reaction, right? And in the gluconeogenic pathway. So phosphoenol pyruvate carboxykinase (PEPCK) and pyruvate carboxylase and fructose 1,6 biphosphatase, and last one is glucose 6 phosphatase. And this key enzymes is very important. It's the final targets of the regulation by hormones, right, or substrates. And this slide sort of kind of overview of how substrates regulate, these 3 key enzymes in glycolytic pathway and the 4 key enzymes in the gluconeogenic pathway. On the left side is glycolysis pathway, right, direction, and right side the gluconeogenic pathway, and 4 key reaction enzymes, right? And you don't need to remember all of this, but some key hormones and substrates, right? So I just take one example, like insulin, right? It's a key hormones, and let's see how insulin regulates, coordinately regulates, the each pathways. For example, insulin can stimulate glucokinase, and the second key enzyme in glycolysis is phosphofructose 1 kinase, right? And also the last key enzyme in glycolysis, pyruvate kinase, right? So insulin basically can stimulate 3 key enzymes in glycolysis pathway, so then can increase the glycolysis activity, right? When insulin levels are high during the fat condition, right? But in the meantime, when insulin level is high, we don't want to gluconeogenesis being active, right? We have enough glucose from the diet, we need to handle it, and we don't need to generate or produce more glucose from other precursors in our body. So gluconeogenic pathway in the fat condition should be shut down, right? Or should be decreased. So insulin can inhibit the key enzyme in the gluconeogenic pathway. Like first one is the glucose 6 phosphatase, and insulin can also inhibit the second key enzyme and also third and fourth key enzyme in the gluconeogenesis. So in general, insulin stimulates key enzyme in the glycolytic pathway and inhibits key enzyme activity in the gluconeogenic pathway. So So overall, the results of insulin action will be increased glycolysis and decreased gluconeogenesis in the fat condition, right? That makes sense. So, and we also have several, the substrates, the intermediates in the reactions in the pathway that can also regulate the activity of key enzyme in both pathways and depend on, which in intermediates or substrates. And I think the fructose 6 phosphate and fructose 1 phosphate and citrate, and I guess mostly, this substrates we're going to talk about later. You probably wanted to remember this substrate, how this substrate regulates the key enzyme and regulates the pathway activities, right? Any questions? So this just gives you the general, idea about the regulation. Now let's get to the detail. And again, this I mentioned, right? The nutrients and physiological conditions that changes the hormone levels in the body, right? In the fat condition, we have increased insulin levels, and during the fastings, we have a decreased insulin level but increased glucagon levels, right? So glucagon-insulin ratio kind of changes depending on, again, the fat fasting condition. And the glucocorticoid and epinephrine levels can also change, particularly during the exercise, right? Epinephrine levels change, and this change, epinephrine also involves the regulation of the glucose and, lipid metabolism as well. So I just wanted to remind you, here I just because we're studying glucose metabolism right now, so we're talking about a regulation of insulin, focusing on the glucose metabolism. But actually, insulin can also regulate lipid metabolism, right? Lipid synthesis or lipid, breakdown of epinephrine as well. So basically, hormone regulates the enzyme, the key enzyme in, again, each pathways, and regulate their activities, and also can regulate their gene expression. The activities basically, regulates enzyme, the allosteric changes, right? So allosteric regulation, and also regulates the phosphorylation or dephosphorylation enzyme, right? We'll talk about later. So protein phosphorylation or enzyme phosphorylation. Have you guys heard about, right? The phosphorylation of an enzyme and regulates the enzyme activity, right? So phosphorylation basically is the addition of the phosphor group to a protein molecules, serum, serine, and tyrosine residues. So when you're adding a phosphor group to a amino acid residue of a particular protein, right? Then you're causing the change of a conformation of that protein enzyme. So then enzyme activities will be changing, depending on whether it's phosphorylation of a protein or dephosphorylation protein. So the serum, serine, and tyrosine are the most common amino acid residues on the protein that can be phosphorylated or can be adding phosphor group or remove phosphor group from the residues. And there are actually two different type of enzyme that catalyze the phosphorylation or addition of a phosphor group to a protein, or dephosphorylation, that means remove of the phosphor group from the protein, right? So phosphorylation and dephosphorylation, phosphorylation is kind of irreversible. You can add a phosphor group to the protein and can also remove phosphor group from the protein and changing the, the activity of the protein or enzyme, particularly here, right? So this, adding or remove a phosphor group can happen very quick, several minutes, even seconds, right? So this is a very quick kind of regulation, right, of the enzyme activity, turn on and off by adding phosphor group or remove phosphor group, right? So kinase basically is an enzyme that can catalyze the phosphorylation or adding, the phosphor group to a protein. And phosphatase is the type of enzyme that catalyzes dephosphorylation or can remove the phosphor group from the protein, right? So when you see some name of the enzyme, the kinase or phosphatase, that tells you how this enzyme, whether this enzyme catalyzes phosphorylation or dephosphorylation, right? For example, we learned one of the very important enzymes, glucose 6-phosphatase, right? So that means you remove the phosphor group from the glucose 6-phosphate and then release the free glucose, right? So that means the catalyze the remove the reaction that remove the phosphor group from a substrate. Okay, now hormonal regulation. Now let's talk about insulin, right, which is a very important enzyme, also called anabolic hormones. That means usually insulin catalyzes the synthesis of some, complex molecules, right? Like synthesis of glycogen, is. So in which physiological state would you expect insulin levels rise? Fat condition, right? So in the fat condition, or sometimes they call a postprandial condition, right? After meal. And what do you expect? Glycolysis and glycogen storage when insulin level is high. Glycolysis would be increase or decrease of glycogen store. Increase, yes. So insulin basically stimulates glycolysis and also push, the glycogen store during the fat condition, right? And what about the gluconeogenesis? The glycogen. Glycogen is, I think, the glycogen synthesis and glycogen breakdown. Oh, sorry, a gluconeogenesis and glycogen breakdown pathway, right? Decrease. So insulin basically decrease gluconeogenesis and decrease the glycogen breakdown pathway activity. And what about the two key enzymes? One is glucokinase. The expression or the activity is glucokinase. Increase, right? Glucokinase is the key enzyme in the glycolysis pathway, or activates, increase or activates. And what about the PAPCK gene? Decrease, right? I heard somebody said decrease. PAPCK, yeah, down, right? Yeah, inhibits or different name, inhibits or decrease or inactive the PEP-CK gene. And insulin stimulates glucose transport into which tissues? Skeletal muscle, right? And adipose tissue, right? So net result about a blood glucose level, a decrease, right? So when we have high insulin levels, and we tend to have a lower blood glucose level, right? You know, insulin push a lot of glucose into the cells for metabolize or for synthesis of stored energy like glycogen. All right, and then the other type of the hormones called counter-regulatory hormones to insulin action. I mean, these hormones basically have opposite function, right, to insulin, and like glucagon, corticoids, and epinephrine. And the glucagon basically increase during the fasting condition, right? So in which physiological condition levels are increased of the glucagon. Fasting, right? So fasting or starvation, and the glucagon levels will be increased, and also epinephrine and glucocorticoid. But again, in the fasting, I think a glucagon plays a more important role than the epinephrine. Epinephrine usually plays a more important, in terms of a glucose metabolism, right? So epinephrine basically regulates more like the lipids metabolism, so break down the lipids, right? We talk about later. And which is the main target tissue of a glucagon? Liver, yes. Great. So liver is the main target of a glucagon. And what about glycolysis and gluconeogenesis? Glycogen breakdown pathway and gluconeogenesis during increase, yes. So increase glycogen breakdown pathway to break down glycogen and release glucose, right, to raise the blood glucose level, and also will make more glucose, during the fasting. And how about PEP-CK gene expression or activities? How glucagon regulates the PEP-CK? What do you expect? Activates, right? Activates or increases, expression of the PEP-CK gene, and so then can stimulate the glucose output from the liver, right? So glucagon does two things to stimulate glucose output from the liver. What kind of two things that glucagon does to increase glucose output? Right? I think all listed in the slide. How glucagon, which metabolic pathway, glucose metabolic pathway that glucagon regulates to increase glucose production from the liver? Anybody? We just talked about it, right? Yeah, yes. Glycogen breakdown, right? And gluconeogenesis, right? So glucagon does two things, right? One is stimulate glycogen breakdown, and the other thing is to stimulate gluconeogenesis, right? And to allow liver to produce a lot of glucose and release it into the circulation. Okay? So net results in terms of a blood glucose level when glucagon action is increase. Increase, raise the blood glucose level, right? So now we have an answer, right? Why, how our body raise the blood glucose level during the fasting and decrease the blood glucose level during the fat condition, because of this very important hormone that are changed during fat fasting condition, then regulates all these key enzymes in the, the metabolic pathways, right? Glycolysis, gluconeogenesis, glycogen synthesis, glycogen breakdown, so then changes the blood glucose level, right? And of course, it also involved in the glucose transporter, right? Insulin also regulates, group 4, right? Okay, so I think I will finish this unit. So I list a couple of, actually not couple, just couple, many questions, right? Points that you need to know for this. For example, how different glucose transporters 1 to 4 are distributed in different tissue, like which tissues has a higher group 4 or which has a higher group 2 or something like this, right? And how different glucose transporter, different tissue play a different role in regulation of glucose uptake, right? And fat fasting condition, or something like this, and more. So hopefully you can, go through these questions if you be able to understand or, to address all these questions. I think it's also kind of, for serve as sort of a study guide, right? Yeah, I've listed here, the substrates that you probably wanted to know how this substrates regulate the pathway activities or enzyme activities in each pathway. All right. Okay, now let's continue talk about the regulation of the glycolysis and gluconeogenesis, right? So as I mentioned, regulation basically involves enzyme activity regulation, right, by hormones and by substrate. And we have overall pictures, right, of the, the whole pathway activities, glycolysis versus gluconeogenesis and regulated by hormones and substrate. Now I wanted to go a detail of each reaction key enzyme, how they are regulated by hormones, substrates, right? So first is the reactions, the key reaction in both gluconeogenesis and glycolysis. As I mentioned, this is irreversible reaction, right? It's the first reaction step in the glycolysis catalyzed by glucokinase or hexokinase, and in the gluconeogenic pathway, and the reverse reaction actually catalyzed by a different enzyme called glucose 6-phosphatase, right? And hexokinase, we know that basically is more abundant in the skeletal muscle, right? And it's different from the glucokinase, which is abundant in the liver and the pancreas in terms of their Km and the affinities or their regulation by a substrate. For example, hexokinase, we basically had the lower Km and high affinity for glucose, and the hexokinase activities is coordinated with GLUT4. That means because the hexokinase is high expressed in the muscle, right? And muscle, we know, also have a high levels of group 4. So the hexokinase usually work together with group 4, right, and regulates glucose uptake and oxidation of through the glycolysis. And this enzyme can be regulated by glucose 6-phosphatase, right? Basically, it inhibits by glucose 6-phosphate, right? And glucokinase is usually more abundant in the liver and pancreas, and has a high Km and low affinity for glucose, right? And this, because of their expression level, the distribution, they mostly in liver and pancreas, and these two tissues and basically express high levels of group 2, right? So glucokinase basically coordinates with group 4 and regulates glucose transport and oxidation. And this kinase basically is regulated by blood glucose level, right? And it's not actually regulated by glucose 6-phosphate, but it's regulated by the other type of substrate like fructose 6-phosphate and fructose 1-phosphate. So this just show you the diagram of the glucokinase activity, how the glucokinase activity is regulated by insulin and the substrate. So you need to remember this, insulin and also this 3 substrate that regulates the glucokinase. Fructose 1-phosphate basically is metabolized from fructose, and fructose 6-phosphate is metabolized from glucose, right? And glucose concentration. So basically, you know that the reaction that catalyzed by glucokinase occurs in the cytosol, right? And this represents the liver cells, and here is the nucleus, and the rest of the area is the cytosol. So glucokinase basically is bind to a protein called a glucokinase regulatory protein, and GKRP, right? So these 2 proteins bind together and then stay in the nucleus. And glucokinase only need to be present in the cytosol, right? Then can catalyze the reaction here. So once glucokinase stay in the nucleus, then this reaction cannot be happening, right? Because of you don't have a glucokinase available to catalyze the reaction. So this sort of kind of regulatory mechanism, to regulate the translocation of a glucokinase from nucleus to the cytosol for the reaction. So the 3 substrate, fructose 1-phosphate, fructose 6-phosphate, and glucose, then you basically regulate the dissociation of glucokinase from this regulatory protein and released from nucleus to the cytosol for the reaction, right? So this sort of kind of regulatory mechanism, and so you know that actually the 2 substrate, fructose 1-phosphate and glucose, usually to promote or stimulate this association of glucokinase from GKRP and released from nucleus to the cytosol. And so this 2 substrate basically can stimulate glucokinase activity, right? Does that make sense? Because you can stimulate the release of glucokinase from nucleus to the cytosol for reaction. And glucose 6-phosphate is metabolized, metabolite from the glucose, right? And this substrate basically inhibits the dissociation. So that means allow the more tightly, bind to the regulatory protein. So what will allow the kinase stay in the nucleus and not release to cytosol, right? So glucose 6-phosphate basically inhibits the glucokinase activity of that reaction, right? Does that make sense? Okay. So, and insulin basically can directly regulate the kinase activity, right? So for the glucokinase activity, and again, you wanted to remember the 3 substrates, the 2 of them actually stimulate the glucokinase activity by, promoting release of glucokinase from nucleus into the cytosol. And the other one, like glucose 6-phosphate, basically inhibit this translocation, right? And insulin stimulate directly glucokinase activity. All right. Let me just finish this. And the second key enzyme in the gluconeogenesis. Oh no, sorry, not second. This is a gluconeogenesis enzyme, right? In the same reaction, but the opposite direction of reaction. So glucose 6-phosphatase is the key enzyme in the gluconeogenesis, and this is also regulated by insulin, right? So insulin basically inhibits the glucose phosphatase and stimulate glucokinase, right? So it's relatively simple, right? Just insulin that involve the let me just think. Yeah, that involve the regulation of the glucose 6-phosphatase, right? So insulin inhibits glucose 6-phosphatase. All right. Yeah, I think I will just stop here, and we have informal writing, right? Oh, sorry. I think we kind of a little bit behind, so we have not talked about this yet, right? The bifunctional enzyme activities. I just want you to write about how glucokinase activities regulated by insulin and like a glucokinase is correct. Okay, so how glucokinase is regulated by insulin and the 3 substrates, right? We just talk about it.